Hello guys, I am Shubham from ShubhVed Tutorials. In the previous video, we have discussed about transposition. This video, we will discussing the examples of the transposition. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. So basically, what is transposition? The different types of transposition we have discussed in detail in the previous video. So examples of the transposition or the transposable elements we call mobile genetic elements. Examples of it are even in this chart. Okay, this is a very easy chart actually, and I think this is very important because all the types of transposition are shown with the examples. Let's see one by one. So first is the DNA mediated transposition. Okay, in this bacterial replicative transposon, first one, right? First one. What is the structural features? Terminal inverted repeats. Okay, we can see here. Terminal inverted repeats that flank antibiotic resistance. Okay, means they are giving resistance to the antibiotic, and transposase gene is there in their structure. Structure means gene elements, DNA. Their DNA, what the DNA contains. Now, what is the mechanism? Copy of element DNA accompanying each round of insertion into new target site. Okay, means they are doing copy, right? And here examples are TN3, gamma delta, Phage Mu, right? Then the second one is again bacterial, cut and paste transposon. Okay, these are cut and paste. These were copy. Okay, this was copy. Now here it is cut and paste. Same here, nothing different. Same is there. Terminal inverted repeats that flank antibiotic resistance, same, as well as transposase gene. And here what takes place? Excision of a DNA. See, from old site it gets excised, means deleted from the old site. That is why it is known as cut and paste, right? We have seen it gets deleted from the original site and gets paste means moves to new site. Okay, so excision from the old site and insertion into the new site. That's it. And here examples are TN5, TN10, TN7, IS911, then TN917. Okay, TN means what? Transposon. Okay. Then the third type of DNA mediated transposon is the eukaryotic transposon. Okay, so these are present in eukaryotes. What they are doing? These are also inverted repeats that flank coding region with introns. Okay, what is the mechanism? Same cut and paste. Excision from the old site and insertion into the new site. What are the examples here? Three elements in Drosophila. HAT, family elements. Okay, then TC1 mariner elements. All these are examples of eukaryotic transposons. Then is the second type, RNA mediated transposition. RNA mediated, first is the virus like. Here, what is the structural features? We know that 250 to 600 base pair, direct terminal repeats. Okay, direct means same direction, not inverted ones. Okay, LTRs are there. LTRs means long terminal repeats. These are flanking genes for reverse transcriptase, right? Reverse transcription takes place, integrase also coding enzyme, and retrovirus like GAG protein. Okay, we have to remember this. Mechanism of movement is how transcription into RNA from promoter. Okay, transcription takes place in left LTR by RNA polymerase 2, followed by reverse transcription. Means first of all RNA will be synthesized, then from that RNA DNA will be synthesized. We saw. Okay, first RNA, then cDNA, and insertion at target site. Right. So here are the TY elements in yeast. Okay, copia elements in Drosophila. Right. TY elements means what? Transposon in yeast. That's it. Now in RNA mediated, the second type is poly A. Okay, poly A retrotransposons. Means they are having poly A stretch, polyadenylated tail, right? So what is the structural features? Again, 3' AT rich sequence and 5' UTR, right? Flank genes encoding an RNA binding. We know ORF1 and ORF2, right? ORF1 is RNA binding protein and reverse transcriptase is formed, right? Same here. Transcription into RNA takes place from internal promoter, target, prime, reverse transcription. Okay, this is a mechanism of transcription of polyretrotransposon. Very important. Target, prime, reverse transcription, and this is initiated by endonuclease cleavage. Got it? Here are the examples: F and G elements in Drosophila, line sign elements in mammals, and ALU sequences in humans. So these are the examples, the structures, the mechanism of movement, all this summarized in this chart. So I think this is a very important chart. Okay. Now let's see one by one some of the important, what we can say, transposons with the help of diagrams. Okay. So this is the first one, IS4 family transposons. IS family transposon. Okay, here the examples are TN10, okay, transposon 10, TN5, right? So what they are having, see, they are having tetracycline resistance genes, okay, in the coding region. Here is the transposase gene, promoter is there, right? In and out, see, it is written in here, if this action this direction, and this is out, right? Then this is the defective transposase gene, okay, defective transposase gene, and here are the terminal repeats. See, these are the inverted repeat, okay, as well as here arrows are in the opposite direction, so inverted repeat. This is known as IS10 left because this is present on the left side, and here right. Okay, so basically this function of this transposon is tetracycline resistance genes will be entering in the bacteria. Got it? It is giving resistance for a antibiotic tetracycline. Now, why this is P in and P out? What is this exactly? Let's see. It is basically a regulation actually. See here. P in this direction, right? So what happens? Transposase gene will be produced. Okay, transposon, see, transposon mRNA. And in this out situation, what happens? Antisense RNA is produced. Okay, antisense RNA is produced. Here is the transposon mRNA, and here is the antisense RNA. Now, this is how regulation takes place. Okay, this is about regulation of IS4 family transposon, right? Then why these arrows are shown, that we have to understand properly. To note, okay, in this direction transcription occurs, then transposon mRNA is produced, and in this direction antisense RNA is produced. Now here is the 36 base pair overlap. You can see here 36 base pair overlap. Now what happens in the first situation, B situation, high TN copy number. Okay, when TN10 is produced in high numbers, okay, then what happens? This will stop the production of itself. Okay, it will block its own production. How? Here, these are the two RNAs, antisense RNA and transposon RNA. They are having a region known as 36 base pair overlap. That region forms a pairing. See, RNA pairing is frequent. So if these regions are paired, translation is blocked. See, translation of transposase mRNA is blocked. So translation is blocked. So no proteins will be produced because its proteins are in high amount. Thus, itself TN10 is blocking its own translation, right? Then in the third part, what we can see when the copy number is low. Okay, here TN10 amount is very low. So what happens here? No blocking takes place. Ribosome will bind to the RNA, translation will be occurring. That's it, right? Here RNA pairing takes place. So the RNA is not free. Ribosome cannot bind. Translation is blocked. In the third situation, no antisense RNA is there and TNT is low basically. TN10 itself is regulating this pathway, right? So the ribosome will bind to the RNA, transposon mRNA, and translation will take place. This is about IS4 family. Now the next type of transposon is Mu1. Okay, Mu. This is present in phage. Okay, this is about, I will write here. Phage Mu. How this transcription takes place, let's see. See, these are the mu A transposes. Okay, binding to the terminal inverted repeats. Right? Two here means it's a tetramer coming. Right? One side two are there, other side two are there. So then after this, what happens? Transposome assembly takes place. Transpososome assembly means rest other proteins will come and see. It is getting separated now, forming a structure like this. Then what happens? DNA cleavage means these two transposes will come together. Basically, they will come together, right? And the structure, like we can say hairpin only, it forms loop like structure forms, and it gives rise to DNA cleavage. We can see here cleavage has took place. Other hand, what is going on? Mu B is there. Mu B. This is mu A. Mu A and mu B on this side binds to target DNA. See, this is the target DNA. It gets bind here. Okay. First of all, mu B is forming a complex, right? Filaments, and then this will attack the target DNA. Now both of these will come together. See, here we can see mu A. Here we can see mu B. They are coming close to each other, and then strands will be transferred. The last step, DNA strand transfer. Finished transposome. Right? So this is a transposome. Transposition has occurred. This is about phage mu. Now this is the final thing. So this is a poly A retrotransposons. So here the best example is line signs. Okay, line sign. So this line from the structure we can see here, this line is having 5' UTR, 3' UTR, right? Then ORF1 is there, ORF2 is there. ORF produces RNA binding enzyme, and ORF2 endonuclease and reverse transcriptase RNA is H, right? In the sign, this is absent. See, only poly A is present. This is common. Target site duplication is common. But ORF1, ORF2, 5' UTR, 3' UTR are absent. So what is the case here? The line is having everything, right? Everything required for the transposition. So this line is a autonomous transposon. Okay, this is a autonomous thing. And this is non-autonomous. This we have to remember. Sign is non-autonomous. So this line will help the sign in its transposition, right? It will give its enzymes to this. Okay, so this autonomous will help the sign in the transposition, right? Okay, and the next best example of this transposition is the VDJ recombination. Okay, VDJ recombination. This we have to remember. What happens here? Antibody production. Okay, we know antibodies are specific against a foreign particle. So this takes place with the help of VDJ recombination. right? This will not discuss here in detail. This comes under a immunology section, right? So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section, and subscribe to the channel, guys, and stay tuned for the upcoming videos.